title that I choose for this is Genome-wide responses to a reverse selection experiment between hot and cold environments in Drosophila melanogaster. So basically, this is the background that we start with. Basically, these two papers from the same institute in Vienna. The authors found here thousands of SNPs associated with hot and cold environments. These are results of Manhattan plots, so which showing the result for the CMH test, finding thousands of SNPs associated with these two environments. These are the results for around 35 generations of selection. They also found that this kind of many, or a lot of different SNPs are associated or correlated with some genes with a relation to the response to the stress, to the hot and cold environment. So what we wanted to do after this part is trying to ask the question about what the effect is of the genetic background in this kind of experiment, and also trying to look at the possibly reverse effect when we took the flies already selected for hot and put it into the cold. So trying to see if there is a kind of reverse effect between these two treatments. So these are, we use the same populations that they use. This is basically the temperature that they use for cold and hot with a 12 hours photo period. It's basically a changing environment. It's not a constant temperature. Changing environment for cold and changing environment for hot. And this is the kind of experimental phylogeny that we got in the end. Basically starting from a base population, doing the selection for hot and cold for around 35 generations, and then took the lines in hot, already selected for hot, and divided in two different lines. One line for continue for the selection in hot for another 35 generations, and the other line for make a different cold selection that we are trying to call reverse here in this part, that is basically a cold selection for another 31 generations. So we have three treatments to compare basically here. The line selected only for cold, for hot, line selected only for cold from the base, and this part that we call the reverse part. There are a line selected for cold, but starting from the lines already selected for hot. So we have basically three important time points here. We did this 3 times, 3 biological replicates, and we choose to have the approximation with a pool-seq whole genome sequencing by Illumina. We used a pool of 500 individuals, a population size of 1,000, constant population size across the whole experiment, an equal proportion between males and females across the whole experiment. So basically with this experiment we try to address 4 questions. The first one is if there is any reverse effect on the SNP selected in hot from the base when we put it again into the cold. The next question is how important is the genetic background in this kind of reverse transition between hot and cold. The number 3 is to what extent the genomic responses for cold are responding also in the reverse, basically comparing these two treatments, because these are basically the same treatment, and the only difference is the base population. This one here starts from the base population without any selection, a specific selection for temperature, and this one here are starting with the selection from the flies already selected in hot. And the question number 2 is, number 4, is how are the dynamics of the chromosome inversion frequencies of Drosophila in these 3 experiments. So basically the approximation that we choose to follow after training and mapping and everything in the bioinformatic work was following a pipeline of the population 2 software. Basically starting with a SNP calling, we come up with around 1.6 million of SNPs for the whole experiment. Then we perform the CMH test and the corresponding Manhattan plots in this part. Then we try to look at the overlaps between the selected SNPs. I mean, how many of the SNPs selected in each of the trimers are common SNPs responding to different environments. Then we try to follow the trajectories of those SNPs. I mean, how many of the SNPs selected here, what happened with them in the reverse part and also in the code. And we also took the advantage of a recent paper here that published a set of SNPs markers that allows you to calculate the frequencies of the inversion frequencies of the inversion chromosomes in Drosophila, and allows to follow the trajectories of those frequencies using a pulsive data without the haplotype data. And finally, we try to perform analysis of the reversibility of selection across the most significant selected SNPs from the hot 37, this generation here. So basically, this is what we got after this approximation. These are the Manhattan plots showing the most important time points in the experiment. As you can see here, because we used the same populations from the earlier experiment, this is basically the same result. We got thousands of SNPs that are responding to the specific to hot, thousands of SNPs specifically responding to the cold, and not too much overlap between the two fragments. But what is interesting and new in this part of the experiment is that the SNPs that are responding in the cold, selected from the base, have not responded here in the reverse part, even when this is basically the same fragment. It's a cold fragment. The only difference is the base, the population base that we use. These are starting from the hot population. So this is this is kind of suggesting that the genetic background is very important in this kind of experiment. So to deal with that, or to try to see how important is the genetic background, we selected the top 2,000 selected SNPs from the CMH test, and we tried to see the overlap between them. So these are the two fragments, the population selected for cold, the 2,000 selected SNPs for hot, and the 2,000 selected SNPs for the reverse part. And as you can see here, there is not too much overlap between the cold and the reverse. It's almost the same overlap that we have between cold and hot. So kind of we are doing a selection for a different kind of environment. And more interesting is that we have a very high overlap and significant overlap for SNP selected in hot from the base and compared with the selected SNP in the reverse part. So suggesting that it's very important this genetic background population that is used as a base population in the experiment. So we then try to look at these 2,000 selected SNPs, trying to follow the trajectories of them in the different time points of the experiment. So for example, here we took the top 2,000 here, responding to the cold, selected from the base, and then try to see what happened with them in the hot and in the reverse part. And we can see here they are responding only in the population selected for cold. They don't respond in the hot, don't respond too much in the reverse. Seems to be here, but it's not consistent across the different replicates. However, when we took the overlap between the 2,000 selected SNPs continually selected in the hot, we find kind of ongoing behavior for this reverse effect. They are responding, increasing the frequencies for the hot populations, don't respond to the cold populations here in blue, and they have kind of negative responses here in the reverse. It's not too much, it's just detectable from the graphics, not very consistent between the replicas, but we think that this is an indication that ongoing changes are taking place in these replicas for this part of the experiment, doing this perverse effect in this part. We find similar results for some of the chromosome inversions here. These are the inversions that we found in our experiment, 6 of the inversions in Drosophila, and we found 2 of them responding differentially to the different environments. As you can see here, the inversion C is increasing in frequency across the hot populations here, and is decreasing for the cold. And this one here, MO, is doing the opposite behavior. It's increasing for hot, for cold, but here in blue, and is decreasing a little bit in hot. And we also find for the inversion C this kind of reverse effect. They are increasing in frequency for the populations in hot, and when we put these populations in cold again, they start decreasing the frequency again. So we think this is a good evidence that this inversion frequency is responding with this kind of inversion effect. This effect was not significant for this one. Looks like it's maybe having the same behavior, but it was not significant, at least with the number of populations that we used. Finally, we tried to perform a test of the reversibility of selection, trying to avoid the effect of the threshold when you choose top 2,000 or top 2,000, 3,000 or 5,000 or everything, trying to look at what happened to the whole genome, looking at different windows. So we try to compare the proportion of the SNPs doing this kind of behavior, increasing for the hot and then decreasing in the reverse. How many, or what is the proportion of the SNPs that have this kind of behavior in the reverse part compared to the hot? We used different windows for this. We got 1,000 of these with that. And always for the 3 replicates and all the windows that we compared, we always find that the proportion of SNPs with a higher reverse effect was always high for the reverse part. So we think that this is a good evidence in the end to say that we have at least ongoing changes with this reverse effect. We found that for the individual SNPs and also from the inversion frequencies. We also want to highlight that the differential response, we got differential responses for reverse selection from between the reverse selection and the cold selected from the base, and that this is due probably to the genetic background effect. And there is a recent paper in the institute that already tests this hypothesis. I want to thank all the people in the lab to make this study possible and all the institutions. And thank you.
Two-part question. The first one is related to policy method. Sounds like you had a high number of lines, like 1,000 lines per line. And so what sequencing coverage you have in line to cover, you have a good representation of the polymorphism?
Yes.
And my second question is, it's about recombination, because heat is known to use to increase recombination. So do you think that the frequency innovations that you observe related to recombination changes?
Okay. For the first question, we have a high coverage in those experiments. After all the informatic work of dreaming and mapping and everything that part, we come up with a coverage of around between 50 to 200 of coverage for each position in the whole genome. So we think that was
it sounds like 80% of the flags were not represented in the picture, right?
Yes, that's right. And for the other question, yeah, we don't think that the hot is increasing the combination. It's just selecting the flag they have or doesn't have the the frequency, the inversion,